So in part two, we're going to look a little bit more at the structure of DNA and the discovery of DNA, and a little bit about RNA. So Watson and Crick, they based, and that's the two guys that you always hear about because they developed a model of DNA, which was called the double helix. But Watson and Crick, one was a biologist and one was a physicist, but they based their work off of the work of Rosalind Franklin and Erwin Kargoff. Remember, he was the guy that said that had the rules about apples and trees and cars and garages are adenine and thymine bond only together, and guanine and cytosine always bond together. But in the 1950s, Rosalind Franklin, she and her work wasn't really recognized in biology textbooks until a lot later on. And now currently she's in your current textbook, and then all textbooks going forward recognizes her work in the discovery of DNA. But in the 1950s, she did X-ray images, and here's a picture of the image that she made. Not really sure how to interpret this image, but she had this really interesting, it looked like an X, the image, but this image was stolen from her desk by a coworker and given to Watson and Crick, and otherwise they may not have, it may have taken them longer to develop the model. There's a short video right here about Franklin, and if you watch it, it's really interesting, but it talks about her work. And then, of course, Kargoff and his A's and T's and C's and G's and those rules, that was in 1949. After that, looking at the work of, okay, adenine and guanine always go together, and cytosine and thymine, Watson and Crick developed this model, and the structure fitting together, not only the chemical molecules and how they realized that what they were looking at is, if you look at Rosalind's X-rays, if you were to look down the center of like a spiral staircase, a lot of times you have the little steps, but then you maybe have a pole that's down the center that the steps are attached to. And that's what this is a representative of, is it was looking down on that structure, that spiral structure. So then Watson and Crick developed their model. Again, he was a physicist and a biologist, and they called it a double helix. So it was described as two long coil strands of molecules. You can watch this. This is kind of an interview about Watson and him telling about how they discovered DNA. But if you're choosing, watch the one on Franklin. I think you'll find it a little more interesting. But they were credited, they were given a Nobel Prize for their work, but also the Nobel, at the time, they recognized Kargoff for his work, but unfortunately, Rosalind Franklin had died, and so Nobel Prizes aren't awarded to people who are not living. And so sadly, she didn't get the recognition during her lifetime for the work she did, but she is credited later, and the value of her work. So here it just shows, why does DNA replicate? If we go back to our chapter on cells, where And I thought I put a slide in here. Yes. So replication occurs in which phase? Now, when we studied in chapter 3, cells, we talked about cell division and mitosis. You had the prophase, metaphase, anaphase, telophase, and cytokinesis is when it was separating. The longest phase was interphase. In interphase, the cell is growing in size and it's doing its thing, whatever it's supposed to do. If it's a pancreas cell, it's making insulin or pancreatic enzymes. Whatever its function is, if it's a muscle cell, it's contracting. So it's doing its thing. During this phase, at the end, once it grows so large, then it goes into a time. This S phase is when the synthesis part is when DNA of that cell is replicated. So DNA is replicated during the synthesis stage. That's when, and in order to replicate, the DNA has to split apart. And so this double helix splits apart. An ACE is an enzyme. So this polymerase enzyme kind of is like scissors and it cuts the DNA apart. And so you have a single strand of DNA here and a single strand here. Then you have the chemicals adenine, thymine, cytosine, and guanine that are in the cell. Those are nucleic acids, and they will match up. And so here I have a new strand, but right here. So this new strand that's forming, when it eventually completely zips apart and all of these nucleotides reattach, then you have two identical copies. And that's what we want to happen in replication, because a replica is an identical copy of itself. This is really important because if there are any mistakes, then those mistakes are going to be in every cell that is reproduced after that. So I really talked to y'all about this before. At the end of the synthesis or the S phase, when replication is complete, there's a checkpoints. And so these checkpoints will check for mistakes. You know, are all the A's with T's and all of the G's with C's? If there's any kind of mistakes, then the cell will not, in this G2, the cell will not divide. And so this is really important to prevent mutated cells from dividing and growing and producing more bad cells. So if there's any kind of the mistakes that happen during the synthesis and the replication of the DNA, then in the G2 phase, there's these checkpoints that check for that. And if it's too bad, then it won't divide. So that cell will then die. Occasionally you have mistakes that slip by the checkpoints. And it's kind of like if you have autocorrect on your text messages sometimes. Sometimes the autocorrect doesn't correct it correctly. And so it's sent with that mistake in it. And occasionally that happens in your cells too. When you have a message, which is the DNA that is replicated, but then it doesn't get replicated correctly. So this just shows again the DNA unzips in the middle. The way it breaks apart like this, there's little hydrogen bonds between these nucleotides that hold them together. Hydrogen bond, it's fairly strong bond, but it's weak also. But then once it unzips, then you have each side can make a new strand. And then those letters will match up, the A's and T's and C's and G's. So the length of your single rings and your double rings, it's gonna, the rungs of the ladder are gonna be the same distance apart. So now you have two new strands of DNA. You have your original pattern and then your new strand that is paired with it. And this little video link shows about the DNA replication. Maybe explains it a little bit more, and then it's maybe a better visual than the PowerPoint. If we look at chromosomes, remember whenever we see a chromosome like this, we know that it's two homologs that are joined together by centromere. We'll say that this is chromosome number 3. Then this is its copy. But if we unwind the chromosome, it's super coiled together, and you keep unwinding it until eventually you can see this double helix structure. Our DNA is very complex, and I'll show you about that later. In the first studies of DNA, they looked at bacteria because bacteria DNA is very simple. But then human DNA, we've got a lot of extra information. We've got lots of A's and T's and C's and G's. This little slide talks about a library of books holds some books for reference only, meaning you can't check them out. But you can, there's some books that you can borrow and take home. But your DNA that's in the nucleus of your cell is like a reference book. You can't take it out of the nucleus. So in order to get information out into the cell, that's the reason that you have RNA. So RNA is a copy of the DNA that can't leave the nucleus. So let me look. This is just talking about the structure of DNA. Always A's and T's and G's and C's. Your 2 purines that are double rings, and your 2 pyrimidines that are single rings. So RNA is the ribonucleic acid, and it's involved in the making of proteins. So we are proteins, plants proteins. So if you're looking at living organisms, a living organism is essentially a protein. So DNA is the pattern that's used to make RNA. DNA is like that reference book in the library that can't be taken out of the nucleus. So what the RNA does is RNA goes in and it gets a copy of the message or what DNA directions are for making certain proteins. So the differences between DNA and RNA is a single strand, where DNA is a double strand. And instead of using thymine to bond with adenine, in RNA, you're going to use the base uracil. So it's going to be A's and U's in RNA. So DNA is in the nucleus. It cannot leave the nucleus. And so to get directions out to the cell of what to do, RNA has to make a copy of the message or the directions. And then RNA can leave the cell nucleus and go out into the cytoplasm to the ribosomes. And what do ribosomes do? Back in chapter 3 when we study cells, ribosomes make proteins. RNA takes the message of DNA to the ribosomes, and then ribosomes can put together the proteins. So the differences between RNA and DNA, RNA has ribose sugar. The RNA also has uracil instead of thymine, and RNA is a single strand instead of a double strand. And the base uracil is also a single ring if we look at the structure. So a couple things go on. Replication is a copying of DNA. A transcription is kind of a copy. If you're texting messages to your friend, you may not spell out all the words of a sentence, you might use the letter U for YOU or the number 2 for 2. So the message, it still means the same thing, but it's not exactly, it's not spelled out. And DNA spells out the messages and the directions, and RNA makes this kind of transcription, this little texting directions, and shortens it somewhat, but changes maybe the lettering, because you've got U's instead of T's, but doesn't change the direction or what's supposed to happen outside in the cytoplasm. So this is called a transcription. Transcription is when you copy a sequence of DNA to make a complementary strand of RNA. So again, it's a message from DNA that's kind of been texted. The meaning is the same, but the lettering is different. The things that recognize where does RNA need to start copying DNA, that is controlled by these polymerases. They show what part of the DNA do you need to copy. Again, if we use the reference of the library, if you go to the library and you want to do a study on, I don't know, grasshoppers, and you go to a world book, you might not look in the A's and the B's, but skip directly to the G's. So you're wanting to look, and RNA needs to do that also. It doesn't need to copy this whole strand of DNA. It only needs the part where the message is that it needs. And so it starts by the polymerase will cause this to break apart, and it signals where the RNA is to start copying, and then it's okay if it extends past that, but usually there's a stop enzyme also. Once the RNA has copied the part of the recipe that it needs of the DNA, then it breaks apart, and then this DNA attaches back together, coils back, and then this leaves and goes out of the nucleus into the cytoplasm. So that's the little bit about RNA. We'll come back and talk about RNA and what it does once it gets outside of the nucleus and in the ribosomes, and how proteins and things are assembled once that message is out there.